we are starting our last module, that is the microbiology. And in this module, in the first half, perhaps you have gone through the cell organelle of the microbiological unicellular, mostly they are one, and how they come to and organism and having some intricate body parts and all these things functioning, the emergency functions of the organism. But here it is a unicellular organism. That is why a single cell, how they operate to make them live in this harsh world. For judging this part about their survival, about their growth, and about their nutrition, how they are getting nutrition, and all the metabolism in a single cell with the smallest organelles, how they perform all the activities is our matter of. Now, what are the requirements, basic requirements for bacterial growth? We are talking about bacteria now only. What are the requirements? Oxygen, water, the pH, the optimum pH, temperature, source of carbon, nitrogen, that is they are the organic compounds, and the inorganic salts like sodium, potassium, sulfur, phosphorus, calcium, magnesium, all these things, as well as vitamins. Now, the obligate aerobic bacteria, that means the recipient of oxygen. The aerobic bacteria encounter the oxygen damage during their growth by producing the oxidizing enzymes. That is the special type of respiration. They use oxygen for producing energy. What is the main function of respiration? We have already gone through this one. Can respiration? Why we need to respire? Why we have to take inhale oxygen? What is the need?
It provides energy.
Can anybody tell? So why energy? How energy is produced?
It oxidizes
Krebs cycle in aerobic process.
aerobic
Yeah.
oxidizes
So,
glucose.
yeah, so oxygenations are given, and in the higher organisms, the red blood cells carry oxygen. It sends the oxygen in individual cells, and there the metabolism of food happens, because we take food and the food is degraded in smaller fragments, producing the energy or ATP as a byproduct. And this ATP is needed to perform, to function mostly all the functions of the body systems, the cell cycle, the cell division, DNA replication, transcription, translation, all the functions, basic functions for living. They need some energy, and this energy is supplied by this respiratory behavior and the cleaning of complex food. Some enzymes take place. They are the peroxidase, superoxidase dismutase, catalase. So they have different functions. Read this one thoroughly. Whenever I will be comparing points has to be noted for exam purpose, not only for exam. Find the cue for memorizing the methods, memorizing the happenings, what is going inside a living cell, a living organ, and a living organism. Now, for memorizing enzyme name, just find out the names who have AAC at their end. All are enzymes. Now, peroxidase, they oxidize hydrogen peroxide into water and nicotinamide adenine dinucleotide phosphate. Dinucleotide, not phosphate. So these are the cofactors and this is the enzyme. So the cleaving of H2O2 into H2O. The superoxide dismutase or SOD, they reduce the oxygen into H2O2 and O2. Catalase, they reduce hydrogen peroxide into H2O2 and O2. The difference between peroxidase and catalase are here O2 is the byproduct and in case of the first reaction, O2 is not the byproduct. Cofactor NAD is the byproduct. Some pathogens, they grow with reduced level of oxygen. Somewhere there is very scarcity of oxygen, but they can grow. Human being can't grow where there is no ample oxygen concentration, but some pathogens can grow. They are called they can be divided into two: facultative anaerobic. They also respire. Through the plasma membrane, and where there is scarcity of oxygen, the respiration happens, but without oxygen. It is called the anaerobic respiration. Anaerobic. Aerobic and anaerobic. Examples may come in the exemplify the bacterial populations. Name 2-3 bacteria which are obligate aerobic bacteria. Obligate aerobic means they must need oxygen to have respiration. Mycobacterium tuberculosis, P. aeruginosa, they are the obligate aerobic bacteria. Now, the anaerobic bacteria, they are the Neisseria type of bacteria which can respire without oxygen. Now, they are of two types. One is facultative anaerobic, means prefer growing in the presence of oxygen but can continue to grow without it. May or may not be. Preference is oxygen, but without oxygen also they can grow. The example is Staphylococcus, Streptococcus, E. coli. The obligate anaerobic means they must need the absence of oxygen. Means they can't grow in presence of oxygen. So bacteria grow by absence of oxygen, use recipient inorganic molecule called fermentation, mostly found in intestinal tract. So in the GI tract, gastrointestinal tract, oxygen level is very low, almost no. And there also bacteria grow. The whole microbiome of our body, it is mainly functioning through the GI tract, and they are obligate anaerobic, means that microbiome bacteria grow in through this experiment, the oxygen or until the medium becomes anaerobic towards the bottom of the tube. The presence in and the lower end you are, the more lower end you are going, the less oxygen is available in this media, and the bacteria which are of
Ma'am, the audio is not clear.
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
just they will be also growing and they're anaerobic, but they can situation somewhat. They can go and strictly anaerobic. They will aerobic and anaerobic facultative,
Ma'am, your voice is going
need for
sometimes. Couldn't able to follow what you are saying.
I will be changing the net. Let me start with my phone. Is it okay?
Yes, ma'am. Now it's fine.
Okay. So, depending upon the process, they can be categorized into these heterotroph, autotroph, or saprophytic. Hetero means they derive energy from breaking down complex organic compounds like fat, tissues, they live on dead bodies. So they are hetero. Auto own food. The human heterotroph. The food to take energy source carbon, oxygen, and nitrogen from the other plant or animal. But the plants, they can fix the natural energy, carbon, nitrogen, oxygen, sulfur from the nature to give the photosynthesis reaction is and all the biomolecules. But here, the autotroph bacteria, they themselves can fix carbon dioxide to make their own food source. Using light energy, photoautotroph or oxidation of nitrogen, sulfur and other elements, chemoautotroph means if light is used for producing food or fixing carbon dioxide, they are called the photoautotroph. If chemicals like the nitrogen, sulfur and other made components, natural components are used for production of energy, they are called the chemoautotrophs. Now the saprophytic bacteria are the populations of the bacteria. They take energy by fermentation or respirations found in nature, in decaying material, soil, water, vegetation, circulations of minerals like things. They just take their energy not from any chemical. 1 minute. Which are already decayed, from there they take their energy. Now, nutrition carbohydrate minerals as in their feet. Bacteria for growth. Mesophilic, psychophilic, and is it okay? Hello. So in the neutrophilic bacteria, the optimum pH needed for their growth is 7 to 7.2. Acidophilic, they grow best in acidic environment, less than 5 pH. Mesophilic bacteria, they grow in certain temperature, that is 40 to 20 to 40 degrees centigrade. Inside the human being, the body system, mostly grown bacteria, that is below 10 degree, they can grow and they is more common in spring, means they grow in high temperature. Now, bacterial growth, how just the bacteria is growing. The bacterial growth mainly depends upon their solution. It is called the bacterial division from one bacteria cell to two identical daughter cells, from 2 to 4 to 8, and so on. The methodology of bacterial growth, it is very important. The name is called the binary fission. They are divided just in the middle of the bacterial cell, and the generation time is 15 to 25 minutes. So fast they can divide and grow, give the daughter cell rise, and the can happen fast. The bacterial strain originated from a single cell can happen so fast. You see how the bifurcation in bacteria occurs. This is the cell wall, and just below the wall, you can have look the yellow dot, not dot, a line, a thin yellow line. It is plasma membrane, and the entangled thread-like structure, nuclear area, that is the DNA area. So how the cell is the cell crosswalk and subsequent cell separates. And from step 1 to step 4, 15 to 25 minutes, within 25 minutes, from one cell, two cells are grown. Of bacteria gives rise to now there are 4 phases. Phase and then the death phase. The decline of their number happens after a certain time. So it is the bacterial growth curve. If questions are asked, what is the component of the curve of bacteria? These are the 4 phases. Life phase, growth phase, not only growth, it is the exponential growth phase. Very fast the growth occurs, and nextly they reach to a plateau of the graph. It is called the stationary phase, and after passing some time, the bacteria population declines and death phase occurs. The measurement of bacterial growth followed by growth of the cell count in nutrient broth, because in the cell plate, if you just streak with a metal streak, already the loop is there in a metal stick, and dip the metal stick into the liquid culture and you streak over a solid agar plate, then you put it in a certain temperature and pH condition, and after overnight culture you can see there are dots, the cell cultures, the cells, the colonies are growth, and the measurement can be taken by counting individual. You just see, it is a loop, the tip of a stick, this metal stick having a loop. You dip down, there are liquid. Suppose in a tube you have liquid bacteria culture, you dip your stick into this bacterial culture, and then in a solid agar plate, you just stick like this, you make a design like this, and incubate in an incubator in certain temperature, and overnight culture will give you bacterial colony formation like this. This is a Staphylococcus aureus after incubation. Can you see responding. I understand whether you are online or offline. Institute net problem is severely faced. Can you hear me?
Yes, ma'am.
Okay, okay. So bacterial growth in the McConkey agar, it is a growth medium. You have strict. Can see when the day when you are sticking with the bacterial culture and through the loop, you can't see anything. But over the culture, you will be having some appearance of bacterial colony growth, and you can count individually 1, 2, 3, 4. From a single cell, the colony will be grown. It is a more vivid picture. At first, the liquefied media, the culture media, the broth, they are poured over a petri dish. They are allowed to dry. After drying, the solid agar plate are looking like this, and you can demarcate different divisions through streaking the uh, what we say, the marker pens. You can demarcate different areas. In different areas, you can streak different populations of the bacteria, and you can have a look after 1 day incubation. The colony will be looking like this. The design, how you move your stick, it will be giving rise the colony formation. Do you have class now?
Yes, ma'am. We have electrical technology class right
Yes,
now.
ma'am. We have electrical class.
Okay, so
Sir is waiting actually, ma'am.
up to slide number 50. Okay, okay, sorry. So I am just taking your attendance and then leave, please.
Okay, ma'am.
Up to slide number 52, it is completed. And afternoon, today afternoon, we are fixing the class. When you decided?
Ma'am, today at 8, I think.
8? No, no, 8 to be sure.
No, no, faster
Who is waiting? Let me stop the recording.
পাঁচটার পর কি হবে?